Hello, everyone, to another episode of Nerd RX podcast. I'm your host, Barkha, and today we are going to talk about a behavioral technique called Grimace. And to talk more about it, we have a very close friend of mine, Rahul, who is a graduate student and also an entrepreneur. Welcome, Rahul, to the show.
Thank you so much for having me, Barca.
Why don't you introduce yourself to our listeners about your education background and what are you doing right now?
Absolutely. So I did my bachelor's degree in cell biology and neuroscience at Rutgers University in New Brunswick, New Jersey. This is where I grew up. And during this time, I worked in several different labs, mostly kind of in the molecular neuroscience domain. And there I actually was very fortunate to have these very formative experiences that helped actually catalyze a lot of my thesis work here at UNC Chapel Hill, where I got to work with some big pharmaceutical companies like Merck and Eli Lilly and had some great mentors at both organizations that helped me kind of think about biology in this scalable space and from an automation and scaling lens in many ways, um, which kind of segued into my thesis work at UNC Chapel Hill um, very nicely. Um, and here I'm a PhD, I'm a fifth year PhD student in the neuroscience curriculum. Um, and I do my thesis work in the laboratory of Dr. Mark Silka, and the lab is broadly interested in autism and pain. There's actually a pretty interesting intersection of those two areas, but I mostly focus on pain. And the, as Barker mentioned, I work on a unique behavioral measure called Grimace, as it relates to measurement of pain in these preclinical models.
Wow, thank you so much for that introduction. So, today's topic we have grimace, grimace in animals. So, what is grimace, Rahul?
Yeah, so grimace, even in people, we grimace too. Um, Darwin actually posited the idea that facial expressions are really important um, for especially mammals to communicate internal states to conspecifics. Um, that's a fancy way of saying people of the same species, especially mammals, we often use changes in our facial expressions to communicate internal states, whether it's happiness, joy in people, sadness, displeasure, also people. Um, and grimace is really what we know about it currently is really a show of pain in some ways, or a reflection of the affective or emotional component of pain in rodents and other mammalian species. Um, Actually, people grimace too when they're in pain. There's uh, clinical tools like the FACES scale that look at changes in the facial expressions um, in humans to determine how much pain they're in. And it's quite interesting because the same way we use grimace scales in the lab, we actually use grimace scales in the clinic for nonverbal um, individuals. So the way that kind of grimace scales work is that in mice, at least, we look at the relative change in different facial features. Depending on the coat color of the mouse, um, we can identify different features that change during pain. In black coated mice, we look at the eyes, the nose, the whiskers, and the ears. And in white coated mice, it's possible to look at those four action units, as we call them, along with the cheeks. And what's interesting is and especially as a new mom for Barca, babies aren't able to communicate with their parents
Mm-hmm.
or loved ones for quite some time. And a lot of the ways that we look at pain in the clinic in humans is through verbal explanation of pain. How much pain are you on a scale of 1 to 10? Can't exactly do that with a baby. So often grimace scales are applied in with babies to identify how much pain they're in because they also change their eyes, their nose, their eyebrows, and other features. So it's quite interesting that the scale that we apply to perhaps some of our earliest members of our species, right, little babies, um, because they're nonverbal. We also apply that same idea of changes in facial expressions to denote the pain state of nonverbal animals that we can't
Mhm.
communicate with, obviously, to understand their pain states.
Right. So is this a new technique? Because from my knowledge, like I had never heard about Grimace. Like you introduced me to Grimace actually, and all I had heard about are the different other pain measurement techniques, which like requires like the paw surgeries and so on. So why don't we use this instead of other techniques? Because this is less invasive.
Yeah, so that's a great question. Grimace is relatively new, and you're absolutely right that it is not very popular at the moment. That's some of the work that me and my colleagues are doing to kind of reduce this barrier to entry for laboratories to use Grimace. Um, you're absolutely right. A lot that historically the field has relied on something called evoked pain measures. So we often apply noxious or painful stimuli to the hind paw or other um, surfaces of the mouse, essentially, and we simply observe withdrawal reflexes. And that absolutely does kind of reflect a component of pain, which is the sensory component of pain, right? So if we think about pain from very meta space, right, you often have to identify a potentially or actually noxious stimuli. That requires, at some level, some sensory processing, right? Like this is a hot surface. That is a sensory like component of the pain response. Identification of this is hot and potentially dangerous. Another component is that is a hot surface and it's going to be unpleasant if I keep my hand on this for too long.
Right.
That's often something we would call an affective component of pain, attaching emotional valence to a sensory or perceived sensory experience. Um, and one other component is like the cognitive component of pain, right? Like now that you have this sensory and emotional information, um, how do you deal with this essentially? Um, and that's actually quite difficult to do, in my opinion, in mice. Um, but from the two easier components to study, which is the sensory and affective, um, a lot of our traditional measures have been on that sensory front, um, where we can apply these noxious stimuli and see how the animal responds with regards to removing its paw from a hot surface or a mechanical stimuli, and the relative latencies that are involved in those tests. That's historically what the field has used. Grimace is relatively new in the last, 10, 12 years or so. Um, Jeff Mogul's group at McGill introduced the Grimace scale in mice, and since then it's been applied to actually quite a few different animal species, including rats. So a lot of the common laboratory animals, essentially, that we use in preclinical research. Um, and I think, one of the limitations, right, so historically has been creating very turnkey solutions to evaluate Grimace. I think there's a lot of commercially available assays right now and systems to study evoked measures of pain. And I think that's quite important, right? When we have standardized automated procedures to evaluate any type of behavior, it helps with rigor and reproducibility. It also helps with reducing barrier to entry, um, excluding financial constraints. These systems can be quite expensive.
Right.
Um, but that just hasn't rolled out for Grimace until fairly recently, and that's kind of like what we do in the Zilka lab. Me and my colleagues have been working on reducing that barrier to entry because up to this point, there has not been standardized behavioral chambers, recording parameters, optical and lighting, arrays that have been developed to really ensure that people are collecting very high quality um, Grimace data. There just hasn't been enough traction in the field to this point to ensure that, and that's what me and my colleagues work on. And kind of the technology I guess we'll talk about today will hopefully shed some light on what those major challenges have been historically and how we've addressed them.
Right. And you have also been involved in making these test chambers, right? And
Yeah,
so tell us more about that.
Yeah, so like I said, my background's in cell biology and neuroscience. I'm not necessarily an engineer or computer science person by education. Um, so when I joined Mark's lab, I had a very unique opportunity when I joined the Grimace team, essentially, to start thinking about what is the best way to make scalable systems to acquire Grimace. And that kind of required me to look at what has been done to this point. And I think I was very fortunate that our lab is a close collaborator of Jeff Mogul's group at McGill, who developed the Grimace scale, as I said. Um, so we had opportunities to have a lot of discussions with them on what kind of works with the current chambers that exist and what doesn't work. Um, the chambers that when I first came into the lab that I saw the major areas of improvement were kind of all with respect to modularity, meaning how can we maximize one chamber so that it's amenable to modification in terms of, for groups that are interested like yours, looking at, neuronal ensembles and populations. How can we make a chamber that allows like a fiber to be passed through, and you can collect potentially behavioral data as well as ensemble data. Another idea was, how do we make this chamber amenable to multiple animal recordings or restraint or any modification in those domains. So, I spent a lot of time essentially starting from, what has been done. And what I was seeing was a lot of folks are using one, big piece of metal that accommodates multiple chambers and they're not modular. And that's kind of the first thing I told you about that I really wanted to improve. So, um, one of the key design elements that I implemented was making all the chambers single units. And you can learn more about that at hypothesis2hardware.com. We can probably include that in
Yeah,
video notes or
of
something.
course.
Um, and that was one of the first things. The other thing was how can we? I noticed that my colleagues would spend a considerable amount of time essentially mounting their chamber, modulating their lighting, and positioning the cameras every single day to make sure that it was in the same place and similar optics and lighting was there. Um, I'm the type of scientist that I like to make my life as easy as possible because I think science is hard enough as it is.
Yeah,
So one of the things I want to do is enhance rigor and reproducibility. And the way I thought to implement that was through basically standardizing the positioning of all of these different features. So, and these different features being, or facets being the chamber, the lighting, and the camera. So if you fix those permanent or semi-permanently in place, you have an opportunity to minimize the amount of day-to-day variance
correct.
in the optics and lighting, um, because certain parameters are pretty well controlled. Um, so that was another thing that we did for the chambers. Essentially, we mounted them to a breadboard. The lighting was affixed to a breadboard. We actually have a new kind of technology that's been developed that reduces the LED requirement, which used to be like, I think, as you know, you've tried
Yeah,
this too,
yeah.
in the thousands of LEDs. Now we can get away with about 14 LEDs. Um,
Wow.
so it's a huge, yeah, that's a recent development that we've been working on. It's a massive reduction in the kind of amount of illumination energy you really need to achieve high quality videos. As you remember, we went back and forth about
Yeah,
now, like we need to
the
illuminate this
lighting.
better. This
Mm-hmm.
and yeah, so that's something else we worked on. Now it's basically just an add on to the original chamber. And then for the camera, my thesis very much is concerned with the idea of developing scalable systems so that we can ask a range of biological questions at a pretty high throughput relative to, it's never going to be like a 1536 well plate, but because I don't think it exists for mice. But we're really approaching relatively medium to high throughput with mice, an in vivo system, which I think is really special because I think that's what the field really needs right now. We're generating quite a few hypotheses across biology right now. Um, from the scalable systems that do exist, which have traditionally been the multi-well plates, which historically have not been amenable to mammalian biology. So, I think Drosophila, C. elegans, those types of systems. Actually, Drosophila wouldn't really fit the well format in all cases, but I've worked on technology that's leveraged C. elegans in multi-well format, but you can't really put mice in wells.
Yeah.
Um, so the system that we're developing is also beyond standardized in terms of all of the positions are affixed of the three key facets, the lighting, the camera, and the actual hardware, the chambers. Um, it's also amenable to scale. So I designed the system such that you could have multiple devices running in parallel. And that was really through some kind of little tricks essentially with relatively affordable computing in the form of Raspberry Pi devices. They're these tiny little computers that are actually quite powerful for a lot of biomedical applications, especially in the behavior space. And you can get some incredible optics there. They're highly modular. I love them. So, we implemented that along with kind of the chambers as well as the lighting to really create, in my opinion, a turnkey solution to acquire and process Grimace data at scale.
Okay. Nice. And are these chambers commercially available? Like people can buy it?
Yeah, yeah, they can. It's through hypothesis2hardware.com. And yeah, they can certainly check them out. We just released a bioarchive preprint for the technology, and there is a beta testing program that's in place right now,
Okay.
where we're providing, or the lab is providing, the chambers as well as the illumination hardware for
Mm-hmm.
no cost. And labs essentially just have to acquire a camera that we found to be amenable for our system. Because a lot of the things I do is very much on the hardware, data collection, and analysis side. A lot of hard work has gone into the machine learning
Mm-hmm.
that's been involved in this technology that really, in my opinion, has been critical for the advancement of kind of what we're trying to do. And um, we have great collaborators in Gurung Wu and SK Park at UNC, the psychiatry department that have really done the legwork for that and have made a really robust and rigorous machine learning algorithm that helps identify those features that we talked about, the nose, the eyes, the whiskers, and the ears, and assign a score in terms of how much pain they're in um, with pretty high accuracy.
Wow. That's great. And would you give an overview of what this experiment looks like? Like from day one, you start your experiment, how long does it take to get the data? Because some experiments can be quite long. So, what are the steps involved for this experiment?
Yeah, I think it depends on the question at play, right? So, um, some of the things that we're interested in is, how well does Grimace detect the presence of pain and its relief? So the presence component, we often use post-surgical models of pain. So hundreds of thousands of people receive surgeries across the world every year. It's fairly common, and there is a significant risk of um, post-surgical pain development in many individuals after surgery, and a considerable effort is made in the clinic as well as in the preclinical setting to better understand the mechanisms underlying post-surgical pain and how we can perhaps target them from from a therapeutic standpoint. So, kind of our experiments as it pertains to kind of that area,
Okay.
they're actually relatively quick. So we can, bring the mice up, um, as I think Jeff Mogul and colleagues have recently published a nice review on this. There are a range of factors that influence pain, um, and a lot of people beyond the Mogul group have discussed this. But you have to be quite meticulous in preclinical settings to do pain experiments as we're learning. And we let the animals habituate to the move for several hours. Um, and then we do our we do a baseline recording. It takes about 30 minutes per animal. And since we have a scalable system, we run this in parallel. We then remove the animals from the chamber. We use our post-surgical model of pain. In this case, it's a laparoscopic procedure. So we make a midline incision at the abdomen, and then palpate the small intestine, suture the animal, and let the animal recover for about 30 minutes. Um, and then we take a post-surgical recording, and that's where we see the grimace response. So, we can process, tens to 20 animals relatively quickly, within a day, very easily, very comfortably. Um, so I think that's one of the advantages too, where really before you would have to do all these, I mean, you'd have to habituate the animals anyway. You'd have to do your pain, you know, uh, paradigm implementation anyway. And then you would have to actually sit there and like take these evoked pain measures yourself. Um, and now it's just you put the animal in a box, you hit go on the camera, and you upload your videos to PainFace, which is the machine learning algorithm site that processes all the data. And in a couple, in a little bit of time, you have your data.
Wow, this is fairly quickly as compared to other behavioral techniques that I use.
Yeah,
So that's a huge plus.
it's it's really a powerful platform, and we're just excited the things that we've used. So in the paper, which is McCoy, Park, and Patel et al. Let's see if I can pull up the citation actually. Yeah, so it's on bioRxiv, and we could perhaps link it in there if anyone's
Yeah,
interested in reading it.
yeah, of
Um,
course.
but the kind of gist of it is that we use a post-surgical model, and our collaborators in Jeff Mogul's group, they use an inflammatory model essentially. Um, and both models showed robust grimace responses. Um, so, with these injectables, essentially, formalin or carrageenan or some of these other commonly used um, chemical or inflammatory models of pain, people could perhaps run way more animals than we do. We have to do a surgery on every mouse. How
Right.
many of these models will elicit robust grimace responses? I think that's for the field to find out, right? We've developed the technology for these questions to be asked, and if some of them do elicit robust grimace responses, it'll be quite exciting to see um, how this technology can be implemented to discover which assays do evoke grimace, because not all of them do. Um, and then how do we find the relief of grimace too? If you give analgesics, how do they reduce grimace? And that's already been known. Our lab has published a paper on that with our, this is our second machine learning approach to grimace. Essentially, there's one done by Alex Tuttle in our lab a few years ago. Um, that was published in, I think, Molecular Pain. Um, but yeah, he also found that grimace is a quite robust measure to find both the presence and reduction of pain by analgesics, and Jeff Mogul's group also has shown this in the past. Um, so those are the kinds of applications we currently think about, and you're right, they are a lot faster than some of the other behavioral
Yeah,
assays that require
a
a
lot of
lot of
training,
effort. Yeah, yeah,
weeks and weeks of training. My currently the team has experiments. I'm still training them, so
right.
it's yeah. So this sounds interesting, and also like you mentioned, it can be used in so many different type of mouse models. Like it does not have to be a surgical model, but as you mentioned, an inflammatory model, and I think that is what I was trying to do with my HIV TAT transgenic mice to see if we see something. But yeah, that is another story. so, so hopefully we'll
Yeah,
get that working soon. So, what would you say are the advantages and disadvantages of this technique?
yeah. I think the advantage is that it's an experimenter-free technique in some ways. So
Yeah,
you can just put the mouse in the box and you can leave the room. And I think there are reasonably some concerns about um, how individual experimenters may differ in these evoked pain measures. In our case, this is not an evoked pain measure; it's a spontaneous pain measure that doesn't require experimenter manipulation. So I think um, there's a lot of value in that, not having an experimenter necessarily evoke any pain response is good. Um, it's fairly automated. So in the past, and we've put this in the paper too, we've actually about the Grimace scale is fantastic within individual laboratories because you just standardize it across the people using the system or scale. What we actually found in our paper, and this is an analysis I did, was that there is a drop off in accuracy of the Grimace scale as you go across groups. And to our knowledge, that had not been reported before. So, where you did your PhD at VCU, there's also another group that's, I believe, using Grimace um, and for pain and analgesic discovery, essentially. Um, and I'd be very curious, all these different groups that are using Grimace, how much fidelity of scoring is there? Because what we found across two different labs, at least, there is a drop off in accuracy. And I think that is a disadvantage with traditional Grimace measures. And where I see the advantage in our system is that one algorithm is scoring all of this. So you don't have individual biases or differences in how Grimace is evaluated anymore. It's a standardized platform. We're using one algorithm on all of these data sets. So I think from a rigor and reproducibility standpoint, this is fantastic. It's a great tool. That being said, depending on the types of applications people want to use it for, whether it's, we think about things at scale in the Zilka lab. Um, Not every lab is going to perhaps want to use this in a scaled capacity. They may just have lower throughput of animals for the questions they're currently pursuing. In that case, uh, camcorders are fine to use. They're turnkey solutions, and we have recommended devices that, along with the settings that we recommend to users essentially for our algorithm. Um, I'll say for the scaled applications, it does take a little bit of computational experience
Okay.
and a little bit of building experience to build these setups. Um, if I could do it, anyone can do it. I'm not an engineer by training or a computer scientist by training, and um, it is quite doable. Um, and I think one of the things that we're learning to appreciate more is just like other pain measures, Grimace is very sensitive to um, external factors. So I think in general, as we think about behavior, just being very mindful about small details and how's the shading in that room? How does the temperature vary in this room? Um, how did we bring up the mice? Was there noise going on,
Hmm.
when we were picking up the mice, or like these factors that sometimes are easy to take for granted or brush off that end up being quite important for behavioral results. And that's
Yeah.
not unique to Grimace. That's like across every, um, if you have mice that had disruptive noise the night before and that disrupted their sleep,
Hmm.
you can imagine that
Yeah.
independent of Grimace, many behavioral measures may be affected by this.
Right.
Um, but that's just a bigger thing to keep in mind for the field. So that's not unique to Grimace. Um, I think one of the other things that we're now appreciating as our technology pumps out a lot of data. So traditionally, a lot of Grimace papers have looked at anywhere from 8 to 15 maybe frames um, per animal per condition, um, which is not a lot.
Yeah.
And we actually run the analysis to determine empirically how many frames you actually need to sample to get an accurate Grimace measure, and it's significantly higher than that. And it just highlights that we're generating a lot of data, and the interpretation of that data also has become an art to a certain degree. Like I said, my major role in this paper has been from an analysis, hardware, and experimental um, kind of perspective. And from the analysis side, it took months to develop pipelines on what's the best theoretical framework to even analyze this massive data set. I think at one point we talked about in the paper, um, we have 6,000 frames from animals that had a sham surgery, the real laparoscopic surgery, and the laparoscopic surgery with an analgesic. Um, and we did some novel kind of analysis on this data set. And it took, a long time to learn what's the best way to approach this big data, which previously just did not exist. Like I
Yeah.
said, people were using anywhere from 8 to maybe 15, 16 frames per animal. And now we're talking about we have thousands, hundreds, if not thousands of frames per animal.
Right. That's a lot of data. Like, I can imagine, like, you need a whole another person for just the data analysis bit.
Yeah. And that's something that we're trying to be mindful of in terms of, supporting the community and analysis, um, as this is relatively new for all of us. This type of data sets have not existed for Grimace until
Right.
now.
Yeah. Wow. And what about the disadvantages?
Yeah, so I'd say that's one of the disadvantages right now. It's like how to analyze the data. So we talk about this
Yeah.
in the paper, um, where, I think the traditional ways that Grimace has been analyzed, we're finding some deviations for how we're going about this. So it, at the moment, does require some computational
Right.
experience and a little bit of data science know-how to use some of the packages we use, as well as, implementing kind of the like artistic elements of the analysis
Right.
in
Right.
terms of order of operations and how you want to go about, because each data set's going to be different, and I think that's going to be a big challenge. The hardware and analysis of the raw data, I think, is going to be very turnkey. If you want to get into scalable applications, there's going to be some engineering computational background people will need, so that's going to be a disadvantage, until we can make that also very streamlined. Um, in addition, kind of how you analyze data for each project is going to be its own unique challenge too,
Right.
um, where we are encouraging people to kind of deviate from the traditional parametric averages approach to Grimace data, and we've kind of moved on to, um, more distribution-based methods in some ways. Um, and that's a disadvantage in terms of if folks don't have the kind of data science background, um, there is a learning curve, but I think
Mm-hmm.
it's an achievable learning curve.
Right. And can you use, like, for now, you are using the system only with mice, right?
Yeah.
And can you use any mice, irrespective of their, like, coat color?
Yeah. So right now the preprint we have is for black coated mice. We're working on a white coated mouse algorithm as well. And the overall goal of the platform is really to generalize to other species.
Okay.
So I'm hopeful in the coming years, our group is able to, and the community and field more generally, can leverage this PainFace platform to uh, develop scales for other species in the lab as well as in the clinic. Automating even grimace measures and, um, you know, perinatal settings in the clinic would be amazing, right? If we have continuous monitoring of our, the youngest people in our populations to ensure that, they're living as pain-free as possible
Yeah.
at those early life stages would be a great benefit, right?
So let's talk about the cost associated with Grimace. Is it something very easy for a lab to set up, or it will require a huge investment?
Yeah, it's a pretty low barrier to entry. Hypothesis Hardware sells all the hardware solutions needed to capture these videos. Um, Also, the camera systems, you could either uh, look at options through Hypothesis Hardware, but also we recommend some pretty budget-friendly cameras where under $200 or so one can acquire these videos, using our setup. And then right now, for the analysis side of things, we're accepting beta testers, so there's no cost associated with this. So it's really a low barrier to entry for most labs,
Mm-hmm.
and it's quickly scalable. So with a
Right.
low initial cost, one can determine if this is something they want to continue to collect this type of information or data from a given project longitudinally. and they can scale up accordingly. So it's quite modular, and you're not going to necessarily be stuck with a massive,
Hmm.
expensive piece of equipment that ends up being, a huge capital and um, large footprint essentially in your lab.
Okay, yeah, that I think is another advantage of it because unlike some other behavioral equipments, this is pretty uh, cheap. And like I was just running locomotor activity the other day, and unfortunately, we just have this one chamber, so I have to wait for every animal and accordingly design my experiment, when to give the drug. And so that requires like a huge time commitment to just run one batch of animals. So I think that would give people more access to the equipments from you. Okay, thank you so much for that. And are there any fun facts about Grimace? This is one of my favorite questions to ask.
Yeah, so grimace, from the literature, there's beautiful grimace scales that are being developed for all sorts of animals, seals, horses, ferrets, and this isn't peer reviewed, but a colleague of mine tagged this on Twitter that capybaras seem to have a change in facial expressions that are associated with um, actually like warm baths, essentially. Whether that's a grimace or not, we don't know,
Hmm.
but I thought that was quite fun.
Oh, so they show grimace when they're happy?
It's so when they're in a warm bath, they pull their ears back, and
Okay.
that could you in mice we consider that grimace.
Okay.
But it also brings the idea that perhaps each animal, especially mammals,
Hmm.
they may use different features to communicate different internal states.
Hmm.
So I think that's quite interesting.
Hmm. Capybaras are cute, by the way.
Oh yeah, they're adorable. Um, and I think what we're learning, even from these like funny little tweets or whatever, that are not necessarily scientific, but I think in a bigger picture way, I think it highlights that the facial expressions of mammals can be quite complex and nuanced, and in many ways, we're still scratching the surface of what they mean. Um, even with the Grimace scale, I was talking about how these unique facial action unit combinations denote pain. Um, when mice are sleeping, they also kind of look like they're grimacing. There is
Hmm.
a nuance that this is an active area of research for us in terms of discerning these different states and which are true to pain and which are just perhaps ubiquitous across other behavioral measures. But um, and like I said, in a more big picture way, we previously just have not had the tools to really examine and fine detail the facial features of mammals and how it denotes rather um, internal affective states. And I think it's just a really beautiful area of inquiry that's only now receiving attention. I'm hopeful that it'll continue to be a fruitful research direction.
Correct. Well, this was quite insightful. And my last question would be obviously about any interesting articles. And I think during the show you mentioned a couple of them, and I will make sure to link that down in the description. Thank you so much, Rahul, for giving us your time and going through Grimace.
Thank you so much for having me. This was a lot of fun.
Thank you so much. And listeners, I will catch you on next week for another interesting episode. And if you have any suggestions about any other technique, please email me at barkha@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.